Hello everyone, and welcome to today's webinar from 3D Printing Industry, the authority on additive manufacturing. I'm Hayley Everett, I'm a technology journalist at 3D Printing Industry, and I'll be your host for today's event, which is Regenerative Medicine and 3D Printing Opportunities, Future Direction. So we have a fantastic panel of speakers lined up for you today, but before I introduce them, a little bit of admin. For those of you who aren't familiar with the Remo platform, the chat function is to the right hand of your screen. So here you can chat to other attendees. I see that some of you have already found it. Say hello to colleagues and contact and form discussions that arise from today's webinar. And to the right of that, you'll find the Q&A box where you can ask questions of our panel and also vote up the questions that you'd like to see answered. We will be taking questions throughout the event. So if there is something you'd like to ask our panel, then please do so through there, and we will try our best to get through as many as we can during the webinar. So if there's anything at all you'd like to know from our panel, then please do use this function. The first hour of today's event will be a panel discussion between our speakers, followed by a networking session. And so when the first hour is up, you'll be directed back to the networking area where you can click around the tables and join in different discussions on what we've spoken about today or about additive manufacturing generally. So without further ado, I would like to introduce our panel, and if they can please turn on their cameras and microphones and join me on the stage. Hello, everyone.
Hello.
So joining us today, we have Katie Weimer, VP of Regenerative Medicine at 3D Systems. We have Darcy Wagner, Associate Professor and Wallenberg Molecular Medicine Fellow at Lund University, and Itedale Namro Redwan, the Chief Scientific Officer at Cellink. So thank you all for joining me.
Thanks for having us here.
So I suppose let's start with some introductions. Katie, how about you tell us a little bit about yourself and your role at 3D Systems?
Yeah, thank you. And first, thanks for having me here today, and looking forward to the great discussion. So I'm, as you said, Katie Weimer. I work at 3D Systems. I've spent most of my career here really focused on 3D printing in healthcare for the last 15 years. And now I'm work on our advanced research and development team focused on our regenerative medicine and bioprinting solutions.
And Darcy, can you introduce yourself and briefly explain your work?
Yeah, so my name's Darcy, and I'm at Lund University, which is in Sweden. And my group focuses on trying to leverage biomaterials and bioengineering principles to both study lung diseases in the lab, but as well as for trying to generate tissue for transplantation one day.
And Itedale, can you tell us about yourself and your role at Cellink?
Yes, absolutely. So I'm Itedale. I'm the CSO of bioprinting at Cellink, and I'm currently leading the team working with bioink and tissue engineering. And within our team, we do develop bioinks, but we also work a lot with the applications and, of course, work very closely to our customers as well. I'm also managing the collaborations, the external collaborations, and so on.
So thank you all for introducing yourselves. I suppose it's good to start off with a general state of play of the regenerative medicine sphere. And I guess, Katie, if you could say to our audience, perhaps for those who aren't familiar with the area, what is regenerative medicine?
Yeah, I think when I think of regenerative medicine, I think of, really a branch of medicine that regrows, repairs, replaces damaged, diseased cells, tissues, or even organs. When you think about regenerative medicine, I think it can include the use of therapeutic stem cells, tissue engineering, and ultimately the production or manufacturing of artificial organs. And I think when we think of bioprinting, really in this nascent space, we think of bioprinting really as that intersection between regenerative medicine and 3D printing.
And Darcy, perhaps you can give some insight into why 3D printing is being used in this field and what some of the benefits are.
Well, I mean, depending on if you're talking about for tissue engineering purposes, depending on what tissue you're trying to regenerate, they can be incredibly complex. And so 3D printing has a lot of versatility as a manufacturing technique in general. It also allows for personalized medicine approaches, which is something that can be either cost prohibitive, time prohibitive. for other sorts of approaches. So I think there's a lot of intrinsic benefits to having additive manufacturing with this sort of tailorability for patients. But there's still a lot of areas that need to be developed and sort of reimagined, since originally 3D printing, either you come from the thermoplastics or solvent-based techniques, with the powder beds. So there's still a lot of work to be done, but I think, all of us that really believe it's more the sort of the concept that we can really imagine that this is something that could be translated to patients one day because of the benefits of the manufacturing approach.
And Itedale, how about you give us a perspective from Cellink and why 3D printing is being leveraged there?
Absolutely. And I think automating the dispensing technology and also having machines doing it basically can give us a higher freedom to include and incorporate artificial intelligence in the future, and also adding a lot of quality control aspects to the process. I think today many of the people say we are happy with manually dispensing and we have very reproducible results, but we are trying to create something that can be applied for the future and also add the complexity that bioprinting provides the customer with. And I think basically at Cellink, we are a technology provider. And of course, we try to work with our customers, not only to offer them what they need, but we listen to them when they're using what we already provide them with to make sure that they get what they really need and what they really are asking for.
About regenerative medicine and bioprinting, it's important to consider things like FDA approval and clearance. And I suppose, Darcy, how about you give us an idea of what these terms mean and why they're so important in this area?
Well, I mean, again, if we're thinking about, something that we're going to try to put into a patient one day, I think the earlier that we think about what that pipeline is going to look like, we can design other aspects into our preclinical type of studies. So a lot of the materials that we're going to use, some of them may have already been put into humans, for example, maybe not, of course, not in a bioprinting form, but some of the materials we might select from might already be approved, but we might not have materials that are perfect. And so we might need to really completely go through that process again. There's also a lot of aspects to consider in addition to just that the materials being, approved. Where is this going to be manufactured? So I think that's something that I definitely think there needs to be a lot more discussion, because maybe we can prioritize different, research efforts. Are these, if we're going to tissue engineer, is it going to be done at every hospital? Are we going to have centralized expertise? Because there's a lot of expertise required for this. So, are there going to be regional hubs or for certain organs? One of the other things, at least here in Sweden, for different organs, you only can get transplanted at certain hospitals for certain organs, because that's where the expertise is. You're going to have to think about the biopsy from the patient if you're going to use the patient's own cells, for example. So I think there's a lot of logistics still to be thought about. And if we can do research sort of that in that mindset, we're mimicking, what might lead to a product that can go into a patient one day. And I know Katie, that they have more experience with thermoplastics right now. And so there's probably some things we can learn from that. But from the bioprinting side, some of this is going to have to be done differently. So maybe Katie can comment a bit more on, sort of the evolution that they've had. And I know that Itedale also, that they've had these, thoughts as well of, what bioinks do we start with? What do customers want to work with? And I know they have a lot of polyethylene glycol, which, this goes into patients, right? So, those are the obvious starting points, but they might not be the perfect solution, to really regrow organs. So I think there's a lot to think about. So it's a loaded question. So I hope I at least started the discussion on that topic.
Yeah, Katie, what's your perspective on that?
Yeah, and great to follow Darcy. I think it's a great introduction to it. And I think, at least with our experience with over the past 15 years of 3D printing in healthcare and being a medical device company ourselves and supporting that, medical device community with several other clearances and CE marked products that are used today, I think we have a perspective from that 3D printing in healthcare and learned a lot from that. And we can pull that into moving bioprinting from, that prototype or that R&D phase up through production. And I think the regulatory environment for 3D bioprinted end-use products is still being formed. The initial guidance from the FDA on 3D printed medical devices didn't even come out until 2017, after several other devices went forth and sort of pushed that forward. There were no specifics in that guidance for bioprinting yet, although I know there's a lot of discussion in the industry about, helping push that regulatory pathway forward. I still do think the major regulated component categories of it's a device, it's a drug or biologic, I think those will still stand. I think we'll see a lot of combination products, especially as Darcy said, when you bring in the cellular component to it, it adds a whole nother level of complexity. And then I loved the comment, Darcy, that you made about point of care printing, or where will the manufacturing, will it be a centralized model or will it be more localized? And I think that, the logistics even of the bioprinting of living tissues with cells will push that point of care printing even further. There's a lot of great kind of basic work being done in the industry today around things like anatomic models, but like you said, those are like harder plastic type materials and even some metals, but we haven't yet pushed the limits of that into the biologic components. So I think that point of care printing, Darcy, as you brought up, is going to be a really important conversation for us to have moving forward.
Have anything to add to that discussion?
No, I couldn't agree more. I think the best way of doing it is by doing it together. I mean, what's really important, the complexity, it adds on. If you go from a single material to multiple materials and then adding cells on top of it, that increases the complexity. But I think important steps have been started to be made in the industry. We just need to work together to make sure that we can provide the value of bioprinting for regenerative medicine in the translational landscape. And this is where I see we should work a lot more together.
Great. So let's have a look at some of the recent developments in regenerative medicine. Darcy, coming to you, how about you tell us a little bit more about your work that you're doing in your lab?
Yeah, so, when I sort of started my lab a few years ago, I really wanted to get into this space because of the reasons that I outlined. Is that, I come originally from the decellularization field, which is what you just basically strip off the cells from an organ and you're left with the scaffold. And I'm originally trained as an engineer. And so I always had problems with how much variability there was just between each, whether even if it was pig or human lungs in the case of my lab that we were decellularized. I just, I thought, if we have to start somewhere different each time, maybe machine learning can help with something like that in the future, but it's kind of a nightmare. So when I was thinking about, the space that laid in front of me and, starting a career and what do I want to devote, a significant portion of my time to, bioprinting was something that immediately, there'd already been some momentum in the field. And so I sat and really thought about what are the areas that need innovation, like most desperately. And I actually think the printers, this is just, my opinion, other people may have different opinions, but, my opinion is that the printers are okay right now. They're maybe are not perfect. And I know, we've got people here that are going to come out with, better versions of printers all the time. But, frankly, I don't know if I'm convinced that those are where the innovations are needed. And this is why my lab really focused on bioink development. And I think one of the other things that we surveyed in the field, at least, or, I did at the beginning, was that a lot of really promising bioinks, they didn't have tissue specificity, so they had a lot of good general properties for a bioink. So, they had really good gelation components, for example. They, you're able to actually print with them. They don't have too much shear force on the cells, things like that. But these are very general properties, and you really want things, to generate organs. You want things that are tissue specific. So what we started to focus on is actually sort of mixing, a little bit of my background with decellularized matrices and taking inspiration from people that had turned those into solutions that you could then print. There's a number of problems with those solutions directly. So as Itedale already alluded to it, and Katie, that, blends and different combinations of inks is something that I'm a big believer in. And so that's where we started. So we took alginate, which is a very heavily and widely used, biomaterial ink, and we mixed it with extracellular matrix from lung in this case. But this platform you could easily use with any organ conceivable, and also from different species. which we don't need to get into today, but maybe a bit on the regulatory. There's some pros and cons in both directions for that. So, so this is what has been the main part of our focus. We are developing other bioinks, which we hope to reveal to the world soon. But I think the main focus that at least that I'm taking is that we really want to design things for specific tissues and applications. I think we need to move beyond this proof of principle and really start to drive towards trying to get functional tissues, which is a general problem in tissue engineering. We've learned a lot, I think, over the last 30, 40 years about how to put cells on foreign materials, but we really have to take it to the next level and get functional tissue. So that's at least a brief introduction to what we've done. So we recently printed, small airways, for example, where we were able to regionally specify 2 different cell types. So you could have epithelial cells on the inside and surrounded by a layer of smooth muscle cells, for example, on the outside, using the same bioink, but a dual extrusion system that you have. You can regionally specify the cells. That's a very quick update of sort of what we've achieved and what we're looking towards for the future to really push the functionality of these bioprinted constructs.
Fantastic. And just a reminder to our audience that you can ask any questions of our panel in the Q&A box, and we will try and answer them as we go throughout the webinar. So, Katie, how about you outline the ongoing developments at 3D Systems and with regenerative medicine there?
Yeah, so we're really couldn't be more pleased with the recent announcement of the Alevi team joining 3D Systems. Really just a fantastic extrusion lab-based platform with installments all over the world. And to Darcy's point, just doing, with people doing just amazing research, and where we're really focused is continuing to push that lab-based, in vitro regenerative medicine application opportunity. And I agree with you, Darcy, that the material development part is really important. We fundamentally as well believe that the materials drive the adoption in the end-use applications of this technology. That being said, I do think maybe the burden's on us as the hardware manufacturers to continue to improve these technologies, higher throughput, continuing to push the limits of resolution with specifically extrusion-based bioprinting as well, and continuing to get to those advanced bioprinting solutions, moving beyond the basic research, Darcy, as you said, to continuing to push towards those advanced bioprinting solutions. So, using the bioprinting solution to create 3-dimensional cellular structures to really imitate the natural tissues, working on, bone, working on blood vessel, and really facilitating that in vitro testing in the regenerative medicine space. Beyond that, we're really excited to talk to the world about our print to perfusion process. So this is sort of a different approach, right? Where instead of what most of the industry is doing today with printing the cells directly, we have a solution where we can print really high resolution, specifically vascular structures as an example. And then secondarily, after the print, perfuse those cells onto the tissue construct, really to create living tissues that sustain life. So this is, I think, a great advancement that can really push the R&D to the next level. And it really allows us to push the limits of resolution, which historically have been a challenge with some of the extrusion-based printing. And I know, Itedale, you know the differences there too as well with your diverse platform of printers at Cellink. So we're just really excited to be a part of the industry and continue to push the limits. And as Itedale said, collaborate, work with customers to kind of get us from this R&D stage. And we're really excited to push that towards end-use human applications as well.
Okay, great. And Itedale, can you tell us about the developments underway at Cellink, perhaps about some of your recent acquisitions?
Absolutely. And of course, I'm very, very pleased to share and welcome our most recent colleagues to the Cellink family. So we recently merged with 2 companies, Matec and Visico, both US-based companies, of course, having their technology, Matec specifically, having their technology spread all over the world. And of course, these 2 mergers gives us, it adds on a lot more expertise to the Cellink group and Cellink family. Although we do quite different things, the end goal is the same. Talking a little bit about what we're doing now, something that has been our mission for a very long time is, of course, to do our best to minimize the animal models used in the industry, and specifically for drug development, cosmetic testing, or any other life science research related question. And of course, that was also one of the reasons that we decided to merge with Matec. But what we are doing in Cellink bioprinting is that we are working very, very heavily focused on showing the value of using bioprinted advanced models that, of course, are adds the complexity to single or simple 2D models, but also adds another dimension to 3D cell culture by having biomaterial. And in addition to that, we use a lot of different printer-related technologies to, let's say, pattern what we print in a specific way or another. This is, of course, one of the biggest mission for the company. In my team, of course, we are working a lot with bioinks. And this is, of course, one of the reasons we've been in touch with Darcy as well. We do see that the printer development, the technologies out there, they are very diverse, they are very high quality, and we can actually get very low resolution as well as high resolution. But what's really in contact with the cells is the biomaterial, and what can really help us get more mature tissues and more advanced models is by really tailoring the bioinks and biomaterials. And of course, that's still a topic and an area where we will continue to work.
Okay, brilliant.
Maybe just a question, if I can interject a little bit to Itedale and Darcy. We all talk a lot about these in vitro models for drug development and testing, yet it seems there's a little bit of a slow adoption to move towards these 3D, more 3D printed complex tissues. In your minds, what do you think is prohibiting the field from adopting that quicker?
I have some opinions. I can go on that from the academic side. I mean, I think the first question, I've done some drug screening projects in my life, and the first question is, who's doing the drug screening? Is this in pharma or is this being done in academia? And you see both. Yes. And so I think that's probably the first question, and I don't have an answer for it, but it's more just a comment that both those things happen. And my lab is also in the space and some other projects of developing new models, right?
Yep.
Our plan is to also be the people that do the drug screen, because there's so much expertise required to handle these models. So kind of you're looking for this really small group of people, in my mind, that are able to generate, at least right now how we envision it, they would need to generate the model on site and perform the drug screening. Yeah. And I know maybe there's some big pharma that's interested in that, but from what I understand, from presentations, it's not that I have some insider knowledge on this, but when I watch other academics give presentations, it's that they are collaborating with pharma who provides the compound libraries, right? Or they do this in collaboration. So I think that's one of the things. One of the other things, and it's, it's not something that I'm not ruining anybody's idea here, I think, but, I think, if we can ship some of these, so there's some, things, maybe can we work out logistics to ship? So maybe you've got somebody who has the expertise in one location to actually generate these models, and then you could ship them to pharma, right? Or to, there's, in Sweden at least, and when I was in Germany, there's national drug screening facilities, right, that have open source sort of chemical libraries. So I think it's because of the necessary, complex expertise required, to do that. And so there's already not a lot of people that can do bioprinting, even if it's for in vitro. And then on top of that, it's an even smaller community of academics that are familiar with drug screening and how to set that up. So that's my perspective on it. And so I think I can only encourage, if there's anybody from, national funding bodies here, you've got to have grants that give enough money, right, to all partners to make it worth their time and investment, because there's just a ton of logistics involved in those sorts of things. And I think, that's something that maybe some national funding bodies are a little bit hesitant, to maybe give money to a drug screen, but this is needed, right? We set up these advanced models and you've got to test it then, and likely at the academic site, or you've got to pay that the technology can be transferred to, to a pharma company that can do the drug screening. So that's my take on your question. I think it's a great question. So that's my take on it.
And Itedale, do you have anything to add?
Absolutely, and I can think of at least two reasons, but I think I'm going to stick to those two. And I do want to say, I mean, of course, we are working very, very closely with pharma, and what we see is, of course, the bioprinting industry is still young. It's matured in the past 5 years, maybe past 6, 7, 8 years, but it's still a very young area compared to the other conventional techniques that are being used in the pharmaceutical industry. 3D cell culture has been adopted a lot, and it requires, number one, it requires a lot more proof of concepts that bioprinted models really work better or even equally good as 3D cell culture models. That's something we need to work on the industry. One team in Sweden is not alone. The academic scientists doing it one by one is not alone. I think what we need to do is really step in there and do, share the responsibility there, because I think people in the biotechnology and biomaterials and bioprinting field, they understand the value of adding extracellular matrix and these molecules already from day one, and not waiting for your 3D cell culture to actually start creating and producing their own ECM. So that we need to work on as an industry. Number two, if you look at the printers out there, and I know we all are working on systems. For example, we as a technology provider, we continuously listen to our customers and create newer systems. But if you look at the people or the scientists or the researchers who really enjoy the bioprinting industry, it's most likely engineers who likes tweaking and fixing and, playing with their G code and changing their STL file and the testing materials, even develop their own materials. While pharma, they really just need a reliable way of creating their reproducible model in high throughput scale, and it should be advanced enough and equally good as the one they're used to, and maybe they've used it already for 5 or 6 years. So I think it's also puts a responsibility on us, on the industry, to work with pharma to make sure that they get the tools they need. And I think, we recently launched a very advanced system which was developed specifically for batch production, for a more high throughput production of tissue models. One of the applications is tissue models, of course.
And Darcy, we have a question here for you. From
I see several questions in the chat, if I'm doing, or in the Q&A, if I'm doing this correct. So maybe tell me which one you want me to answer that come at me.
So I have seen that alginate can have really good and tunable mechanical properties and gelation, but it's usually a sugar-based ionotropic gel. Doesn't this make it quickly degradable?
Yeah, so it's a great question, and I think I actually want to take maybe a step back, and then I can answer that specifically for alginate. But I think the question you have to ask is, are you trying to 3D print something that is a bioartificial device, or are you trying to print a living piece of tissue that's going to remodel over time? So we absolutely, from my view, we want something that is degradable. So we want something that cells can remodel, that can integrate. If you think about some types of patients, for example, young patients, this is actually a problem in organ transplantation in general, is sourcing of those types of organs. So this is in particular for pediatric. It's potentially a really great opportunity where there's not as many organs available, but then that organ has to grow with the patient. And so you absolutely need something that is degradable. And in fact, in our paper, this was one thing that we feel that was leveraged. So if you just Google my name, you can look at one of our recent papers where we did some subcutaneous implantations of our bioinks on the back of mice, and we saw extensive remodeling. And what I mean by that is we actually saw new extracellular matrix deposition from actually the host animal by infiltrating cells. We saw new blood vessels growing into those constructs. So to Katie's point, vasculature is one of the big challenges. So you could either print it, or you can have a material that encourages vascularization. into the graft. And so then you would have a vascularization or vasculature derived from the host. So I think conceptually, it's important to understand what is your target. And my belief is that for the applications that I'm going after, you want something that can be remodeled. Now, you don't want something that is too quickly remodeled. So that's something that, we are trying to look at and address. And there's another question later about methacrylation. So I can address that sort of now because it goes together. So you can, of course, methacrylate alginate or acrylate it. You can do that to a lot of polymers, and that can induce further stability. But then, you have some other complications that you need to look at. And again, I think, from a timing perspective, you want the amount of new material deposited to match what's being degraded. So if you have a mismatch in that area, then you're going to have a mechanically weak. We are not there yet. And again, we started with alginate as a proof of concept, but definitely there's going to have to be modifications. It may not even be ultimately the perfect material, but that's at least the sort of the concept that we are going after. So it needs to degrade. That's a property. But I think Katie, in some of their applications, they don't want degradation. So I mean, maybe she wants to talk about that. Since it's not just on the bioprinting here, on 3D printing, also that there's some applications that you don't want it to degrade really. So yeah.
Yeah, Katie, do you have anything to add to that?
Yeah, I think Darcy brings up some great points, and at the end of the day, it's very application specific, right? What you want those materials to do, once implanted, really depends on the application. And certainly, just going back to the core of kind of 3D printing in healthcare, there's lots of other materials like titanium, that have been implanted in the human body and clearly don't degrade over time. But when you look ahead to the future, I mean, I think we can ask ourselves, in X years, are we still going to be, implanting things like titanium at the rate we are now, or are we pushing forward more to what, a term they use a lot in surgery is replace like with like. So as we do have to, if we can't repair and we do have to replace diseased or damaged tissue, can we move forward with some more biomaterials that go from a 3D printed construct implanted, and then the body as the bioreactor remodels that actually into living tissue, and hugely important, as Darcy said, and in pediatric patients where the tissue truly has to grow with the patient. So yeah, lots of great science and lots of great examples of using some more traditional manufacturing materials in healthcare. But I think that's why we're all so excited about this industry and about the impact it can have on patient care by moving forward to replace like with like, pushing the limits of regenerative medicine and bioprinting. I think that's what we're really all after here.
And we also have another question, which is aimed at you, Darcy, but anyone on the panel can answer. With your recommendation of the need for advances in tissue-specific bioinks, what regulations should be considered when developing bioinks for commercial value in medical devices?
Yeah, so it's a really good question. So I hope that I understand the gist of the question, but I'll try to just give my thoughts on this sort of space. But I think, and again, because I come from the decellularization field, or another term for this is acellular scaffolds, and some of these are used clinically right now. So there are companies right now that make allogeneic derived decellularized extracellular matrix products for skin, for example. These are clinically used. So I think that there's a really good sort of clinical track record with those sorts of materials if they're processed in the right way. We've got some benchmarks in those fields, for example, depending on how you derive that material. But things like sterility, things like biological activity, I mean, there are some of those concepts which are there. One of the other things that's difficult to think about, how exactly it's going to be because of the commercial value, but regarding, it's also on the regulatory side. Xenogeneic, if you would want to use animal-derived products, for example, maybe there's some other regulations that you might think about. I think other areas that, it's regulated to some extent, but it's also it's regulated more, from what I understand, on a national or cohort basis, and you can look at inspiration, like Katie said, when you talk to surgeons. But some of these things, if you think about organ transplantation in general right now, of course there are regulations around this, right? But it never goes through a commercial entity per se, right? It's hospital to hospital. So there's commercial entrance into different points of the process, and that's regulated. At least in Europe, there's discussion of ETMPs, which this I think will fall into this, which is advanced therapeutic medical products. And so, I think there's other spaces which are going to inform, but I think it goes back to our discussion before, where is this going to be manufactured? And I think that that's, something that ultimately we don't know. We just don't know the answer to that yet. And it may be different depending on the application of how that's done. So the regulatory space will need to evolve with that. And I hope that the regulatory space evolves with the people developing the technology and implementing it.
And Darcy, maybe just one thing to add to that great answer. I think it's a nice time to plug some of the work that's being done to create standards in the industry. And I know, for example, ASTM is working on a standard for extruded bioprinting material. IEEE is working on a standard for bioprinting software. And ASME is working on a bioprinting standard for the extrusion hardware. So, as these standards are being derived, I encourage the community to join and participate. We need the experts in the fields to be writing and helping develop these standards for the industry. It's just the beginning, right? As Darcy and Itedale both said, in this kind of nascent industry, going from R&D to more regulated products. But a good time to plug the standards and everyone helping contribute to those.
No, that's a great point. And a lot of them already exist, right, for biomaterials,
Yep.
applications. So I think we can run a lot more of those, when we get a little bit closer. A lot of those I think can be they're already written in such a way that, independent of the manufacturing method, afterwards you can test sterility, you can test cytocompatibility, all those things. And so I think, hopefully there's no surprises scientifically, and hopefully we can just implement those things quite straightforwardly. Because otherwise we have to create everything that will cause lags and delays. But yeah.
And I suppose this brings us on to the next part of our webinar, where we discuss some of the challenges faced by 3D printing and regenerative medicine. So Itedale, perhaps you could tell us from a technology provider perspective what challenges Cellink has faced.
Yeah, I think this is not for Cellink specifically, but in general for the industry. I think we already touched on the topic of having the appropriate materials to move translational. I mean, creating models in the lab for R&D purposes, I think we are there, and we are getting close to proving that it's applicable to many different tissue models, but it's really having the right biomaterials of the right properties, and of course validated, following the standards, and then of course getting that approved by the FDA and similar bodies in different areas of the world. This is maybe one of the biggest challenges we have in front of us. And I think definitely this is something that we all face in the industry, and I don't think it's company specific at all. I mean, it's really what we all have in front of us. And I see there are amazing science going on in the industry, where people are working with recombinant materials, people are working with already FDA approved materials to create bioinks. And I think the technology, I mean, it's there. Now we really need to focus on getting it into the clinic and having the standards creating, as Katie said, we have the US based organizations doing all of the work. Similar standards are being created in Europe as well. And I see that this is happening all over the world. And I think, so we're moving in the right direction, definitely. And now we need to get those materials approved, because they are what will go into the clinical applicable to be translational in the future.
We have a question here on materials. Today, are there general printing properties when printing with biological material that are similar to other organic or even synthetic materials? So perhaps you could shed some light on that as well.
Of course, there are several different things that you have to refer to when you're developing bioinks. Of course, many of the hardcore biotechnologists, they focus a lot on the printability, the resolution you can achieve, the shape fidelity, and how well the material cross-links and how stable it is and for how long. I think, of course, all of those are important, but what's even more important is how well do the cells survive in the material, and how well do they act as they should. Is it a cell type that is supposed to proliferate and then differentiate and then maturate? Or is it a material that does not proliferate but maturate and behaves metabolically in a specific way? I mean, there are many aspects, because in the end of the day, we can create amazing materials by mixing left and right in the lab, but it needs to fulfill the purpose. And that's also, an expertise that the industry, of course, has. I see these amazing material scientists doing that work, but it's really keeping the focus on not only resolution and shape fidelity, but the combination, and achieving all the points is still a little bit challenging, but we will.
And Katie, perhaps you can give us an idea of some of the challenges that you faced at 3D Systems with 3D printing and regenerative medicine.
Yeah, I think when we look forward, into the kind of the future of this space, I think one thing that we're really excited to focus on is, helping support the scale up from R&D to production. It's something we've lived through in the 3D printing in healthcare space with traditional manufacturing of traditional 3D printing manufacturing, having printed, over 2 million devices, that are either implanted or used in surgery today. And I think that scale up is really important. When you think about these new and nascent technologies, we really haven't had the opportunity through, these products on the market to really test and push these through from R&D to production. So thinking about how you characterize this process, how do you do risk mitigation with little production experience? How do you, what process parameters do you control in a bioprinted solution once it's in that regulated environment? Everything from software workflow to part placement and orientation to making sure you have consistency from build to build. How do you verify the output of a 3D printed organ, for example? So I think this production scale up is a challenge, again, industry-wide we all face. And if, as we do push to more, devices in use on the market today, devices or combination products or biologics on the market today, I think it's really important to also understand the supply chain back to kind of inks and materials. Do we have a steady supply chain to support, a manufactured product that's on the market? We in the industry, can we provide hardware that's reliable and consistent? And so I think talking about this production scale up as we move from R&D to more in-use products, I think it's really important. And to Itedale's point, I think it's very collaborative, both with industry, both with researchers. We have to be thinking ahead, when people like Darcy and her lab are working through this, how do you take the solutions that they're working on and scale them and think ahead to how we scale that into an environment? Again, not necessarily for the commercial opportunity there, but so we can take this amazing technology and help, apply it to healthcare to really improve the lives and improve the treatment for patients. So I think, like Itedale said, it's a real community effort. And one of the things 3D Systems is really focused on is bringing that scale up experience from our 3D printing in healthcare into this bioprinting community. So we're really looking forward to facing that challenge moving forward.
And speaking of the community, we have a question here from our audience. Should tissue engineering and bioprinting move into the open source space? Could these fields benefit from open source collaboration the same way that traditional 3D printing has? What are your thoughts on that?
So maybe I can take that one. So everything that my lab has done has been open source. So we used open source printers. So we actually did a modification of a MakerBot printer. All of our files are deposited. We used extruders that were deposited by Adam Feinberg's lab with some modifications because we also used the fresh printing that his lab developed. So I think it has to be this way. There's another question in the chat about how I've assembled my team. And I think without some of these things being open source, we could have gotten there. It just would have taken us longer. So I think we have benefited from at least a lot of things being open source. And so we try to give back and contribute to that same, effort in the same way. We've got some other things coming out, some other modifications, and we will deposit the 3D printable files so that somebody on the other side of the world can just download it and print them. So I think that we shouldn't hang on to many of these things. There's just a lot of work to be done, and we can't do it alone. So I'm fully in favor of that. With that being said, we have to protect ourselves also, right? So I mean, when innovation happens, this is the world that we live in. We also have to protect various aspects of it. But I think there's some things that if it can be made open source, we should do that. So as much as possible, and also until we really know. It's one of the other aspects of truly open source when I refer to it as that. I don't want to work with a software that I can't modify every single thing right now because, I want to be able to modify everything because I don't know what design or what, choices that, I might have to make. And I want that freedom right now. Later, it might be that, we really converge, as a field on some things and then you don't need that. And then maybe it gets in the hand of biologists, or again, if it's at point of care, like they don't need to make those choices. But I think at least in this early phase, the more that's open source, the better, so that we can just, run instead of all crawling, to redo the wheel at each place.
Yeah. And Darcy, I know we believe in that as well. I know the Alevi by 3D Systems extrusion platforms are all open. And also some materials that are distributed, and the group puts together really amazing protocols. So if you are new to the industry, you can pick that material, pick that, software setting in that printer and get some amazing prints right out of the box. That being said, it's also very open to allow for that material development. I mean, we have to keep these systems open for that reason, especially, that everybody's talked about, to continue to push the limits of materials. So I agree. I think it's a really important time in the industry to stay very open and very collaborative.
Yeah, and I think that goes for, us as well. We haven't locked up our ink cartridges or the accessibility of the materials. In addition to that, there are many platforms available to anyone on the field to log into our BioVerse, for example, and just download the G-code or STL file with the appropriate protocol. And I do agree we should keep the field open. There's only one thing that I truly, truly, truly respect, and it's the publications that the scientists in the academic field are working on. Of course, we have to be patient and wait for them to really publish their data before we can translate it to the industry. And this is something, of course, we continue to encourage, because it's by the peer-reviewed publications that you get a broader community to believe in the data. But that also slows us down a little bit. But
it's really good you bring that up, Itedale, because I think one of the things, at least, I can comment on that. I think a really good thing about the pandemic is there's been a huge push to get research out as fast as possible. My lab's been involved in, to a limited extent, but to some degree, right, because we're a lung lab of COVID-19 research. And so, that is really a trend that I think is here to stay. And so if you deposit on, for example, bioRxiv or any chem archive, whatever it might be, you can deposit those files already. And I think, for my lab, we want to be very careful before we make that step that we really have validated. So I think that's our only hesitation. But, for many things, when it makes sense, we are posting on bioRxiv, for example, since that's the space that we're in. And once we're really sure. So once we're really sure of the results, we publish everything open, because then, most of the time, then the peer review process at that point is really about refining and making sure that the message is delivered well to everybody, and that maybe if there's some other experiments you haven't thought of. But, I think it's very rare that we would post something that its head would get flipped, it'd get flipped on its head, or we would be totally wrong at that point because we've been using it, the advanced. So I think that's something that, these preprint servers has really helped, to get information out faster. But I also agree that, we need to, the peer review is an important step to control the interpretation of results, to make sure that we're not over-interpreting, but it can give, some access to ideas and information sooner while still protecting, our identity that idea came from us, because before the equivalent was in a presentation format like this. There was no written record, the innovation or the advance wasn't maybe attributed to my lab. So So I think that's the really nice thing about the preprint servers, is that you retain that, and I know that there's some industry, I've seen some industry authorship now on some of those as well, which I think is a great move as well. But as soon as all the authors are confident, that they've made an advance, like, let's get it out there, and then whatever journal it lands in, it lands.
And I guess off the back of that, the kind of cross academia and industry kind of partnerships, do you think we need to see more of those going forwards within this space?
Yeah, I can maybe answer on that. Absolutely. Today, I was even sitting in a Finnish society, a Finnish network, where they actually brought together scientists and companies who were sitting and talking specifically about the challenges we have in this bioprinting industry and biomaterials industry. So I see this is, of course, key. And there are several networks. Even in the US, we are actively part of networks and, really working closely. I mean, the expert, I can say, we are a technology provider. We have knowledge, we know what we're doing, but the actual experts are the ones doing the translation in the clinic, but also the scientists who are doing the basic research. And it's only by listening to them, working with them, that we can ensure that what we're doing is the right thing. Of course, you should dare to be brave and predict what is the future and provide that and throw it in the hands of beta users to get their feedback. But we truly, truly believe that it's only, we can only push the limits by working really, really closely with the people who are creating the technology, doing the research behind it, but also the clinicians and the application scientists in the industry who wants to use it and apply it to their real life problems.
We have another question from our audience. How about using other 3D techniques such as DLP and not just focusing on bioprinters? Katie, perhaps we'll come to you and on that one.
Yeah, there's a couple of the DLP systems on the market today, and certainly the print to perfusion system that 3D Systems is developing is DLP based. And I think, that DLP technology is what we're going to need to push the limits of resolution in specifically in the tissue fabrication and even organ fabrication space. So we're actively pursuing that. And I do think, when you get to DLP, there's some different challenges, with cell-laden bioinks, that you have to think about versus that secondary perfusion. But really the DLP for the high speed, high throughput, and high resolution, I think is going to be a really important advancement moving forward. So we're really excited to continue to share that with the community. So that is a very active development in the space, and there are a few other systems on the market today that certainly Itedale can comment on from her portfolio.
Yeah, and we have a great partnership with Volumetric, so we actually offer a DLP printer. We also have a partnership with Crelis Biologics and also have light-based, amazing resolution printers. So we offer the spectra from very, very high resolution to more high throughput, higher speed to the extrusion, which gives you the bulk printing and dispensing. And I do see that the combination of all of these technologies, in addition to microfluidics and perfusion, is what is needed to bring us to the next level. But also, let's not forget about the biomaterials and the bioinks, because it's only those are the closest in contact with what it is we're creating.
And I suppose looking forward now to the potential of 3D printing within the regenerative medicine space. Darcy, perhaps you can predict what may happen over the next few years or what you're expecting to see in this area.
I think that there's been some really exciting, and I'm going to use a word that I hope I don't offend anybody, but there are simple tissues, if I put it that way. So I can appreciate that every tissue has its challenges, but there are some tissues that are simpler than others. So skin being one, for example. And so I think, you might see some of these things start to enter clinical practice, some of these more simple tissues in the next few years. And I think that, we'll also start to lay the groundwork for some of the more complex ones, which we just, this is not a matter of just being able to physically print them. We've got to be able to figure out how long do we have them in a bioreactor, for example, ex vivo? How long is that process? How do we get, the different cell types that are in there? So, a tissue by definition is multiple cell types. So how do we get them there? How do we transplant it? Do we transplant with, some sort of mechanical support or other sort of intermediate support in the clinic? So I think there's a lot of unanswered questions on those things, and there's maybe not even animal models that are sufficient to address some of those, or you have to move into large animal models to be able to really mimic the human clinical scenario. So I think you're going to see some simpler tissues, from personalized approaches. I would guess that the first widespread one would be skin and burn victims, because if there's a way to custom generate large sheets of, tissue, or have a bio ink that can facilitate, skin regeneration, it's a huge need. In the acute scenario, you could generate it very quickly, especially if there's no cells needed. And there's been some success, with some acellular constructs before. So that would be my prediction, but I think, we are still a long ways away. I think there's been a lot of hype in the field. So I think we've made a lot of really good progress, but to actually be able to generate tissue for transplantation, long way away. To the points that both Itedale and Katie have brought up, I can only hope that, there is more opportunities for collaborative work with some of these bioprinted models, because I do think that there are some distinct benefits, but there's a number of challenges, right, that have to be overcome, for those sorts of collaborative efforts to work. But I think that would be the more immediate future, right? That you could really see, whether it's for personalized medicine or more broadly just drug screening in more biologically relevant models. And that could enter, as soon as sort of there's support, I think financial support in place to really prove that some of those models work, and to start that's kind of ready to go if the pieces can be put into place. So that would be my prediction.
Sure, and just quickly, we'll go to both Katie and Itedale on what you think are the potentials going forwards. We are nearly out of time, so we can keep it as brief as possible. Katie, let's go to you first.
Yeah, I think bioprinting's potential in healthcare really is in, either improving or accelerating product development, or in, these end use applications. And I think, for us we're really blessed to be a part of an amazing partnership with United Therapeutics and their subsidiary Lund Biotechnology. And I know, Darcy, you're working in this area as well, but we truly do believe that with this development program, we will be able to manufacture a transplantable human lung, that can help, curb the challenges that we see in transplantation of organs. So, we believe that as an earth shot, not a moon shot. We do believe that, in the coming years, that instead of someone suffering from, a debilitating organ disease, they could instead, in, some window of time, receive a transplant for their new 3D bioprinted lung. So we believe that the day is coming for manufacturing of something even as complicated as solid organs. Again, we really believe in that, and so grateful to be a part of this partnership with United Therapeutics to make that a reality.
Great. And Itedale, any final thoughts from you?
Yeah, no, I couldn't agree with the other speakers more. I mean, this is an amazing industry. And I do honestly believe that we will change the future of medicine and how the healthcare will look like in the future. I mean, already today we can create disease models, in vitro models that can minimize animal models, predict data and results much faster, more reliable, and more reproducible. And in the future, hopefully, we will be able to print tissues and organs to save those many, many lives waiting for organs for transplantation.
Okay, so I'm afraid that is all we have time for in this part of the webinar. We will now go to the networking of today's event. So if you haven't had your question answered, I can see there's still quite a few in the chat that we haven't managed to get through. Please do stick around for the networking session, and hopefully we'll be able to get those answered for you. I just want to say a big thank you to our panelists who have joined me today and treated us to such an interesting expert-led discussion on this topic. I've definitely learned a lot, and I'm sure that our audience have too. So thank you all very much for joining me, and see you in the networking session.
It was really a pleasure. Thank you.
Thank you.
Thank you.